I will start immediately. The topic is about epigenetics and pregnancy. And you already might have noted this slide this morning in the introduction. This is a number of adults with diabetes worldwide between the years 1980 and 2014. So it's a tremendously increasing figure, which is really worrying, especially as it represents a heat map somehow with the regions with the highest number of adults with diabetes in red, and the blue ones have lower numbers. And the highest number is in low-income countries, such as Africa or Southeastern Asia. So this morbidity is extremely, is a very high burden, not only in rich countries, but also in developing ones. So this is what happens at adulthood. If we look now at what represents the prenatal nutrition, it's simply a surrogate, which is good or bad, but it's birth weight. The distribution of worldwide distribution of birth weight with the same colors. The highest birth weights are in red. Countries with the highest birth rate are in red, and the ones with the lowest in blue. And the distribution is quite similar. There might be many explanations for this, of course, but I would think that what Conrad Waddington called the epigenetic landscape might be one of these explanations. Indeed, Waddington coined the term epigenesis a long time ago, before we sequenced the whole genome. And he depicted development as trajectories, starting with the sphere, the stone sphere, which can roll down differing slopes, and depending on the valleys or the hills it encounters. And these trajectories represent typically the fate of a stem cell, which is differentiating into differing tissues, but it may represent as well trajectories of life, which can be designed very early in life. And the sphere might represent, let's say, the intrinsic potential of the cell or of the individual, so let's say its genome, for example, while the environment and the effects of the environment can be represented by the landscape. So very early trajectories of life can be chosen during development under the early influences of the environment. And this is a key concept of epigenetics. This concept is likely to explain a different concept, which is that of developmental programming. More or less, it could be understood with a similar metaphor. And to understand it, two very simple studies. On the left, the growth of rat pups with a very simple intervention, a short period of undernutrition during 3 weeks, but at differing times during life. On the right graph, undernutrition has been imposed to the pups between the 9th and the 12th week of life. It causes a decrease in growth, a loss of weight, but when nutrition becomes normal again, the rat pups start catching up growth, and they come back to their corridor, so their growth corridor. While if the intervention is performed very early in life, there is also an alteration in growth transiently. And when nutrition becomes normal again, the rat pups start growing, but in a different growth corridor. So this is how an individual or can, either an animal or a human, can be reprogrammed according to the early influence of the environment, which includes, of course, nutrition, and especially early nutrition. So it's a process by which a stimulus applied during a critical period of sensitivity, which is early development, has not only short-lasting effects, but lifelong effects. And this translates into the findings by the group of David Barker, very well known, who showed that in a cohort of human adults this time, the mortality ratio due to coronary heart disease or the rate of type 2 diabetes and other long-term outcomes correlate with birth weight. The lower the birth weight, which means the poorest the intrauterine nutrition during pregnancy, the highest the risk of dying prematurely due to coronary heart disease. This programming effect is an important effect. It's not a marginal one, because here in the thin cohort, in the general population, the prevalence of hypertension is shown. And according to the current weight at adulthood of the subjects in the cohort on the x-axis and to the birth weight. And so the effect of the birth weight, which simply varies here between 4 and 3 kilos, more or less, which is somehow within the normal range of birth weight, of variations of antenatal nutrition within the normal range. And the effect of the weight at adulthood, of course, overweight is associated with an increased risk for hypertension. Both have comparable size of effect. So this factor may have a real importance and not be simply kind of a curiosity of development. It means that the baby develops according to the signals he or she receives from the environment, and these signals, the baby takes them as with a predictive value to regulate and to set up its regulations over life. So this is indeed what epigenetics can do. And here is represented what epigenetics means. The epigenome, which is which represents all the changes, the imprinting, which is performed at the molecular level on the genome, is just an additional layer over the genome, which introduces a very important diversity, even higher of that of the genome, as there is an infinity of epigenetic, biological, molecular imprints on the genome. These changes are simply methylation or changes in methylation of the genome superficially, or in histones, modifications of histones, or non-coding RNAs. And they represent, they are produced under the effects of early nutrition or early exposure to changing environments, such as stress or exposure to toxicants, and especially to contaminants. The epigenome then regulates the level of activity of the genes in the genome. It doesn't alter the gene sequence, but it can upregulate or downregulate important genes in our regulations. For example, in metabolic regulations, the setting of our blood glucose value or of our blood pressure. And this may last for life and can be even inherited through several generations. So we can understand the epigenome between the genome and the proteins which are translated, which are a translation of the genome. It is flexible, dynamic, while the genome is fixed, and it explains developmental plasticity, which means how one single genotype can develop into an infinity of phenotypes, of different phenotypes. So this is a representation of the molecular mechanisms, the three basic molecular mechanisms of epigenetics, which are, again, DNA methylation and histones modifications, modification of the chromatin, and non-coding, especially microRNAs. And to show you how epigenetic changes are important and how early nutrition is important, a good example is in bees. A colony of bees is characterized by the fact that only one of them has a capacity for reproduction. It's the queen, which is a larger bee, and it lives longer. But this bee receives a special nutrition brought by the other bees during its early development. It's a royal jelly for a long time. So the capacity for reproduction is acquired in bees. It's not in the genome or in a chromosome. It's environmentally acquired thanks to epigenetic changes. Finally, what is important is that epigenetic marks, which translate the influences of the environment, especially early in life, because a stem cell, coming back to the landscape of Waddington, when a stem cell commits itself to become a neuron or an epithelial cell, as it bears the whole genome, it differentiates by silencing part of the genome which is not needed for this fate, or upregulating another part of the genome which is useful for the fate of this stem cell. So development is an epigenetic phenomenon per se in nature. And then what is important is that early acquired traits, which during this particularly sensitive period of development, can be inherited as well, as if they were genetic traits, at least for several generations. And this is, for example, this happens, for example, when a baby is born to an overweight or obese mother or a mother with gestational diabetes, which is associated with overweight and quite frequent. This brings a very important overnutrition to the baby in utero, not only glucose, but all nutrients, possibly even postnatally. And this acquired trait then affects the next generation. And on animal models, it's very easy to reproduce a transgenerational inheritance, which may explain the speed of this pandemic in obesity and type 2 diabetes worldwide, which is observed. It may be one of the explaining factors, as there is, of course, a leveraging effect of the cycle of reproduction. And if each pregnant woman can transmit an acquired trait to the baby, and this is reproduced over generations, there is an amplifying effect which can be especially interesting to address in terms of prevention, as this is seems a window of risk, but it is also a wonderful opportunity for early prevention, to act early at the beginning of life, which is the topic of this session. So various conditions of pregnancy, which can be normal, variations within the normal range in birth weight, in fetal nutrition, or pathological ones, such as here being born to a mother with gestational diabetes with a high birth weight, or having a fetal growth restriction, or being born preterm with a low birth weight, they have very similar consequences. Epidemiologic studies and follow-up studies of at-risk babies show us that they are exposed to the same risk on the long term. Very differing initial conditions, very similar long-term consequences at adulthood, the increased risk of cardiovascular disease and metabolic diseases. Very similar mechanisms. I have no time to detail the mechanisms, but they are strikingly similar despite opposite context during pregnancy. And here is a single example I would show you as angiogenesis, which is the development of vessels, is altered in preterm-born infants. And this may be one of the mechanisms of later hypertension. And these changes are associated with an accelerated cell senescence of endothelial progenitor cells in these babies, which are the cells responsible for the development of the vessels. And this accelerated senescence is associated, as we found in this study, with a deficit in the sirtuin-1 gene expression due to a modification in blue of one of the histone, which is close to the promoter of this gene. So there are epigenetic imprintings, for example, of being born preterm. Of course, we would be happy to know what happens with this imprinting later in this experiment. We need to wait a few decades, but we hope to be able to know this. Another example on how the early environment affects pregnancy, this is in Gambia, very nice study, which shows that the methylation of specific genes in infants at 1 year of age reflect maternal nutrition at the moment of conception, especially if it was during conception happened during the dry or the rainy season, with different methylation of specific metastable genes, which show the importance of maternal nutrition. Of course, nutrition changes according to the season of conception. And finally, it is possible to check, to approach the gestational age of a baby, not only comparing with early ultrasounds, dimensions of the fetus performed during pregnancy or the last menstruation periods for the mother, but studying simply the global methylation of DNA can tell us with an error of 1 week the gestational age of the baby. And in older people, it shows it is related to the biological age and can explain, at least in terms of association, why some people at the same chronological age are in good shape and others suffer really more of aging. So to end, this is a comprehensive view of what can be developmental programming, the importance of early nutrition and early life environment over the life course, which starts from left with this famous 1,000 days between conception and the second anniversary of the baby. We know today that preconception is extremely important as well. These are let's say, the programming factors which change the risk for later disease or for health, which is a chance, of course, either with variations within the normal ranges of environment and nutrition or in pathological situations and long-term consequences to the right with the increased risk of non-communicable diseases which may follow. And what is important is that this underlines the importance of early prevention of chronic diseases at adulthood. Acting early makes sense. Working with mothers, with parents-to-be, with infants may be more efficient than addressing the whole population, especially when it comes to counseling and to actions of public health, with much higher returns on investment, acting early, even from an economic point of view. And so we have these multi-generation cycles, which are represented here, with a number of factors on which we can act to avoid these suboptimal trajectories, acting early on nutrition, exposure to stress, exposure to toxicants, counseling future parents or parents to be, pregnant women, acting on breastfeeding postnatally, and this is an opportunity, rather, I would say, than more than a problem. Thank you for your attention.
No. Wait a moment. Thank you very much for this wonderful presentation, which confirmed that being a pediatrician is the most beautiful thing all over the world, so we can save the world. The paper is open for discussion because we have several topics. There. Here.
Thank you very much for your topic. My question is, epigenetic is reversible or not reversible? Because it's important to know if it's transmissible by generation.
You're right, I should have mentioned this. It is reversible in molecular changes are reversible. But the consequences, if there are structural consequences, then they may not be reversible. This is, for example, the case when we consider the nephron number endowment of an individual, because it's a capital in the number of nephrons acquired for life prenatally. If it parallels birth weight. The higher the birth weight, the higher the number of nephrons, the better the health of the kidneys, let's say, for life. So if we have a definitely too low number of nephrons, then the consequence of an epigenetic change might be irreversible, while the molecular change itself can be reversed.
Professor Philip James?
Thank you very much for your talk. You stated explicitly that epigenetics is inherited, but you then, in your descriptions, always seem to imply that the inheritance actually came as a result of that babe becoming a mother, and then, in the course of its pregnancy, handing on things. But some people have claimed that the actual genetic changes at one time can be intrinsically inherited, even if extraordinary preventive measures are taken with the mother, so that the mother is not actually appearing to program, in the classic sense, the subsequent pregnancy. In other words, is the inheritance a direct genetic thing or a repeated epigenetic process?
Well, this has been studied mainly in animal experiments. It seems to be a direct inheritance because in the first generation, it may be a direct effect of the maternal exposure as the baby is in utero. But this can be inherited over several generations. And it can be inherited also from the father, especially through micro non-coding RNAs in the sperm, for example. There is only one study in humans. It's in Sweden, which addresses this. We have few human data, but in animal models it's quite clear. We don't know if it is definitely inheritable for an infinite number of generations, but at least for three generations, it has been demonstrated as a true inheritance of acquired traits.